Welcome back. Hope you enjoyed yesterday's learning. Today we are going to discuss some important terms that we will use in this chapter invariably. The first one is genes. So, what are genes? They are functional unit of inheritance. And for the first time, this term was given by Johansen. So, Johansen was the scientist who coined the term genes. Genes are functional unit of inheritance, and for the first time it was coined by Johansen. So, that means before Johansen it was called by some other name, and that was factors, and this factor was named by Mendel. If you remember, biologists were not able to correlate factors when he said some factor is responsible for these kind of characters. So, factors was named by Mendel, which was later named as genes by Johansen. What are genes? Genes are functional unit of inheritance. Next comes the alleles. What are alleles? These are forms of genes. Generally, in one gene we have two alleles, but it is not necessary that we will always have two alleles. Sometimes more than two alleles may be there, and we call it as multiple alleles. We will discuss this multiple alleles later when we discuss blood group. So, at present we will talk of only alleles in a gene where only two alleles are present. Okay. So, two alleles are present in one gene, and who coined this term? Batson. He is the scientist who coined this term alleles. Alleles now, if a gene has both the alleles, as two alleles are present in one gene, if a gene has both the alleles as similar alleles are there, they are known as homozygous alleles. That means both the alleles are of same type. But before this, let me tell you, when we write alleles, how do we write it? When we take any trait, contrasting character, if we take the two traits of it is labeled by, for example, we take a character that is length of a stem. So, yesterday we saw that length of a stem may be either tall or dwarf. If it is either tall or dwarf, one of them was dominant. I told you we will discuss dominant today, and the other was recessive. So, when we write for alleles, what we do? We take the first alphabet of the dominant, that is, allele will be written as capital T for the dominant character. For the dwarf character, we do not take the first alphabet. Instead of that, we take the lower case of this dominant trait, and that is small t. So, what are the two alleles in a gene? Capital T or small t. Both of them may be present together in a gene, that is capital T or small t. If they are present in this way, we call it as homozygous gene. We can take some more examples. For round seed, we have capital R, and green seed we have small r. These are homozygous genes where both the alleles are of the same type. In case of heterozygous gene, if we take the example of the same, in case of heterozygous gene, both the alleles are of different type. This is a heterozygous gene where both the alleles are of different type. Let us take few more examples. Capital R, small r, capital G, small g, capital Y, small y. This is yellow color of seed. Do you remember I had told you we generally confuse between green color of pod and yellow color of seed because we have same colors on both the sides. This is round seed, right? So these are different types of heterozygous genes. Had it been homozygous, what it would have been? Capital Y, small y, small y. For this, capital G, small g, small g. For this, capital R, small r, small r. For this, we had already seen capital T, small t, small t. These are all heterozygous forms, whereas these are heterozygous genes, where both the alleles are of different types. Then phenotype and genotype. If the character is visible morphologically, we can see it from our eyes. Somebody may be tall, somebody may have red color flower, something color, height, position, these can be seen from our naked eye. These kind of characters are called phenotype, which are morphologically visible, and this tells us about the genetic makeup. For example, a plant is tall. This is phenotype. But if I write capital T, or capital T, small t, small t, these are genotype. What phenotype will be expressed? This will be tall, this will again be tall, and this will be dwarf. So these are phenotypes and this is the genotype. So something which is visible morphologically, that is the phenotype for an organism, and the genetic makeup for that morphological expression is the genotype. This is for tall and dwarf. Cross. Cross is simply reproduction of a plant, that is male and female reproduce together, and that is known as cross. In case of this, we can have, when we study a cross, we can study one character at a time. One character, for example, height of stem. So this type of cross, where we study only one character, is known as monohybrid cross. Mono means one. If we study two characters at a time, it is known as dihybrid cross. Di means two. Let us take an example. Capital R, capital Y into small r, small y. Which two characters are we studying? The shape of seed and color of seed. So we are studying two characters at a time. So if we study this way, it is called dihybrid cross. We can study more characters also. We can have trihybrid cross also. if we study 3 characters at a time, it is known as trihybrid cross. For example, RRYYTT into RRYYTT. What are the 3 characters that we are looking here? Shape of the seed, color of the seed, height of the stem. We are considering all the 3 characters. So, what is a cross? Cross is reproduction between 2 organisms. Filial. The offspring produced after reproduction are known as filial. We designate it as F1, F2, F3 depending upon after which mating it has been produced. If it has been produced after first mating, we call it as F1. If we have a cross where F1 is considered, then the generation is called F2. Similarly, we move ahead. So, the offspring produced is known as filial, and we designate as first filial generation, second filial generation, third filial generation. Selfing. Suppose we are studying any character. We have taken 2 traits. This is symbol for male, this is symbol for female. I am writing P for parent. This will produce 2 gametes, both are of the same type. This will also produce 2 gametes, both will be of same type. For gamete I am writing G. Now these two, if these two combine, we have you can see everywhere we are having the same type of genetic makeup of an individual, that is the organism. So, this is first generation F1. And what is selfing? If we take one male from this and another female from the same F1 generation, that is capital T male, capital T small t female from F1 generation, this is called selfing. And then in gamete we will have small t and capital T. 2 different types of gametes are produced, capital T and small t. So, we will have different types of organisms produced in the F2 generation. Sorry, this is capital T, capital T, small t, small t, small t. So, this is what is selfing. When two offsprings produced in the F1 generation are taken to form a cross for the next generation, that is known as selfing. What is pure line? When we do cross between 2 plants, and this cross is done for 5 consecutive generations, we obtain a plant which will always give us the same desired character, and if we get that kind of plant, we call it as pure line. If you will have cross between these 2 type of plant, we will always have capital T, capital T. Even after selfing, we have same plant, so you are going to get capital T all the time. So, if 2 plants are crossed for 5 successive generations to get plants which will produce same type of characters one after the other, that kind of plant is known as pure line, and generally homozygous genes are obtained as pure lines. either capital T or small t. So this is pure line. So this was all for today. Do revision of all the terms, because we will be using these terms very frequently as we move through the chapter. So have a great day. Keep learning.